we'll have right now our opening talk of the hackathon. Will be the last talk, and we'll introduce the Friendzymes. I want to welcome Isaac Larkin. He is a postdoc scientist at Abbott Labs, and he is a DNA wizard, actually. And he is behind a lot of interesting projects like free genes, Friendzymes, and other interesting stuff in general. And he could explain more if you want, but welcome, Isaac Larkin.
Yes, thanks, Isaac G. And Chaz, once again, really excellent talk. Fantastic to have you. And I think that you will enjoy some of the rhymes and echoes from your talk to mine. So let me just share my screen real quick. Let's see, desktop 2, share. Let's get over here. All right, everybody see my screen? Yes.
Yep.
Okay, fantastic. So I'm going to talk to you today about some of those things that Isaac G mentioned, free genes, Friendzymes, and the work we've been doing towards building an open and distributed bionet. But first, we got to start with why. So, the story we like to tell about biotechnology, and particularly synthetic biology, this new discipline of treating biology and engineering discipline, is that it's a story of rapid growth and of world-changing potential. So, we are an IGEM team in the 2021 IGEM competition. IGEM got started in 2003. That was basically the size of it could fit in like a small classroom back in 2003. And then at least as of 2016, it grown to thousands of students a year, this big thing. And so the story we'd like to tell is, look at how much we're growing. Look at how this field is developing. This is Rob Carlson is an expert in analyzing the bioeconomy, tracking its contribution to, for instance, the overall U.S. economy, U.S. domestic product. And what he shows, at least as of 2012, is this sort of exponential growth from the 1980s onward to 2012 in the share of the economy that was made up by biotechnologies. Like, look at how it's growing. It's going to change so much. Just give it a little bit more time. A couple more decades, it'll be great. But the problem is we're on a rapidly approaching deadline. So the IPCC, that's the Intergovernmental Panel on Climate Change, says that we need to keep global warming to 1.5 degrees Celsius or lower if we want to have a decent chance of averting like irreversible and catastrophic climate apocalypse within our lifetimes. In order to stay below 1.5 degrees C, in order to have a better than even chance, better odds than flipping a coin of staying below 1.5 degrees C, we, the entire world, have to cut global carbon emissions by 50% by the end of this decade, by 2030. And we have to achieve carbon neutrality, net zero by 2050. And then we need to pull gigatons of carbon out of the atmosphere every year for the rest of the century. So that is what we need to do. And we're moving in the wrong direction. There was probably a dip of like 7% in terms of global emissions during the colossal pandemic. But still sort of the total consumption and the burning of fossil fuels of various kinds is increasing in the world. And if you look at the list of largest companies by revenue on Wikipedia, you find that something like 4 or 5 of the top 10 are oil and gas companies, moving in the wrong direction. So we need a massive economic, technological, and political mobilization starting right now if we're going to combat this climate crisis and be able to build a good future for ourselves and our children and our future generations. Biological technologies can help there. Interesting. I think I've skipped a slide that I made. I had a slide in here about how technology is insufficient and we need work at the level of policy. So I'll just preface this slide that I'm about to talk about how we can help by saying technology alone, not going to do it. All you have to do is look at like the COVID-19 pandemic for that. Technology did its job in terms of developing vaccines faster than anybody thought was going to be possible at the beginning of the pandemic. We got to clinically approved mRNA vaccines within a year of COVID-19 kicking off, but we're still 3 years out from vaccinating the entire world. That's according to the guy who heads the Serum Institute of India, the largest vaccine manufacturer in the world. And a big part of that is because we did not do good policy around the distribution of the means of vaccine production. In particular, the developed countries are still hoarding the IP related to vaccine manufacturing, particularly for the newer and the more effective mRNA vaccines. So you can have the technology and you can still fail at the level of policy if you don't have political transformation and political mobilization at the same time. But all that being said, technology is insufficient, but it is necessary. And biological technology can help to build a good future and combat the climate crisis in a number of ways. Replacing animal agriculture, replacing the petrochemical industry, basically anything to do with atoms. Like if you need energy, you need like electrons and joules and things like that. Clean energies like solar power, wind power, those are pretty good for those. You don't really need that much in the way of bioenergy, except for maybe a few things like things that can't be electrified, like jet aircraft and rockets. Those things you might need bio-based alternatives for. But anything to do with atoms, building material things in the living world, biology is like a 4 billion-year-old advanced alien nanotechnology that's like powered by the sun and draws down carbon dioxide and turns it into food, medicine, and materials. And like that is clearly would be the basis of any advanced and sustainable human civilization. So these are the things that biotechnology can do: replace animal agriculture, replace the chemicals that we get from fossil fuels and petrochemicals, and create microbial alternatives to carbon-intensive industrial processes, like creating nitrogen fertilizer and cement. So we need to accelerate this bioengineering revolution. We need to do it now. We need to build that resilient and carbon-negative civilization in 10 to 30 years, or else. We need advanced biological technology everywhere and at every scale. And for what it's worth, we're going to need those things whether we actually build the resilient carbon-negative civilization or whether we don't. Because if we don't, then the world's going to become a lot more chaotic, and supply chains for things like food are going to become an issue. So you're really going to want to be able to manufacture food, medicine, and materials local to where you are, whether that is where you are right now or where you need to move to in the future. And so biological technology going to be needed, and we're going to need a lot of it. We don't have enough bioengineers. We don't have enough biotechnological productive capacity around the world right now to achieve this transformation. Just like Will was saying, as Chaz quoted him in his talk, we need 1,000 times more capacity. We need 1,000 times more bioengineers. And how do we get there fast? Well, we can start by looking at what are the barriers. So, bioengineering and synthetic biology have been difficult and expensive, and that's one of the reasons that it hasn't grown as quickly as it needs to. And so one of those main barriers is just the cost of doing it. It's hard to learn to be a bioengineer. It's hard to set up a bioeconomy because this stuff is more expensive than other things. DNA sequencing and synthesis, they've gotten much cheaper than they were 20 years ago. Like the cost of sequencing has dropped by like 10,000-fold since 2000. The cost of synthesis has dropped by something more like 200-fold, or maybe 40-fold. I'm trying to remember my numbers. But it's still the case that, if you look at the sort of the sequencing company that dominates the market, that manufactures sequencing machines and makes up 90% of the next generation sequencing market, Illumina. Their instruments cost tens of thousands of dollars at minimum. Their smallest instrument, the iSeq, $25,000. Their biggest instrument, the NovaSeq, $1 million plus. That's not accessible. That's not democratized. And if you look at the cost of DNA synthesis, so if you try to analogize biological technology or bioengineering to like software engineering, the synthetic DNA, writing synthetic DNA and assembling it into new things is like, writing computer programs. Each gene is a little subroutine. And imagine if every time a software developer was going to prototype a new subroutine in their computer program, they had to pay $150 to do it. Software engineering would not have grown at the rate that it has. And so, this barrier of the cost of synthetic DNA is still a major one, even even though, it has dropped significantly over the last few decades. So, $70 from the cheapest synthetic DNA supplier for a synthetic gene, 1,000 base pairs. And even from the, currently available nonprofits like AdGene, which is this academic repository for, a whole bunch of different plasmids that have been developed in different research labs around the world. It still costs $60 to get a plasmid from them. $60 for a single biological subroutine. Not great. And so that's the cost barrier. There's also this barrier of creative monopolies, or what industry likes to call IP, intellectual property. Patents. A thing that keeps me up at night is, we need all of this advanced technology deployed around the world in the next decade or two, and the time to expire for a single patent is 20 years. So the time it will take for the patents that are registered today to expire will be like the time span during which we need to transform the material base and energetic base of human civilization. That's a real problem. These IP barriers are a hindrance to accelerating innovation and distributing technological innovation. In particular, this is a problem for synthetic biology, because with synthetic biology, the way it generally works is you take these standardized synthetic genetic parts that do particular things. This part makes the bacteria glow green. This part turns on gene expression during arsenic, in the presence of arsenic ions. Put them together, you get a synthetic circuit that'll detect arsenic by making a cell glow green, that sort of thing. But those standardized parts, so many of them are individually patented that, if you tried to put together a complicated device with many of those parts, the time and money it would take you to go and get licenses for each of those individual parts that you put that together into that useful functional device is prohibitive. It would prevent you from actually going out and scaling that whatever useful device you've built and sharing it with the world beyond an academic lab. And this extends not just to patents, but also just to the sharing of the physical material of biology. Unlike with software where everyone can sort of download code directly from the internet, you actually have to mail like physical atoms to people of like cell lines and literal physical DNA. And most of the resources for biotechnology, the physical cells and the DNA, are kind of locked up in these things called material transfer agreements, which are basically these contracts you have to sign when you when a researcher sends you something that they want to share with you. And because most researchers are at academic institutions, or most researchers that would share something are at academic institutions, and most academic institutions have all of their intellectual property, all of their materials. They share them under this thing called the Uniform Biological Material Transfer Agreement, which is nice if you're a researcher. Basically it says if you're a researcher, you can use this thing for research. But if you're not a researcher at an academic lab, the UBMTA really restricts what you can obtain. It basically says you cannot modify and redistribute the thing that you receive. So you can't share it with your friends. You can't commercialize it. So if you found a useful part and you wanted to get it from somebody and then sell it to a community that hasn't had access to it before, couldn't do that if that part was shared under UBMTA. So this is just another major hindrance to like what people can do and how they can actually do useful things with biological engineering. And this extends even to the competition that Friendzymes is now a part of, for IGEM. Through even to this year, the Registry for Standard Biological Parts. So this is the International Genetically Engineered Machine Competition's library of wetware that all of the teams throughout the past 20 years have compiled and contributed. It's a kind of a biotechnological library of Alexandria of all these nice, cool and useful pieces of DNA that people can put together to make cool and useful biological devices. And they don't really, the registry kind of exists in a legal limbo. They don't share it under a material transfer agreement, but they do basically make teams who use the parts from the registry sign an agreement that they won't redistribute those parts to anybody else, and that they'll basically just contribute what they build back to IGEM. So it's sort of this walled garden where there's all this cool stuff going on inside IGEM, and it just sort of stays stuck in there. As I might talk about a little bit later, thankfully that's going to change next year. So there's that barrier of the creative monopolies of the IP, and then finally there's the expertise barrier. It takes a lot of time and money to become a proficient bioengineer. If you're not, for instance, if you don't have access to automated liquid handling robots, you have to develop the manual skills of like moving small volumes of liquids back and forth over and over again, very consistently, keeping track of your samples and things like that. You have to learn how to make sure that you don't contaminate your samples with, just the microbes that are on you or in the air, how to use sterile technique. You have to figure out how to make sure that you're running your experiments and keeping track of the time so that, an incubation step doesn't last too long, or, because that can ruin your thing. And you also have to, remember how to keep track of where your samples are and all these types of things. And in the absence of, systems of, low cost democratized automation and sort of software for managing that type of data and workflow, that takes basically years to develop the skills to do. And so all of these are sort of difficulties, but how can we draw inspiration? Where can we draw inspiration from to see the path that we could go down to solve these problems? So if we look back to the 1970s, how did people make it cheaper and easier and faster to do computer engineering? So back in California in 1976, Apple Computer releases their Apple I. Their first personal computer was one of a few little personal computers that popped up right around that time, and it cost about $3,000 in 2019 dollars, and it cost electricity to operate. Now it had a pretty high skill barrier because it didn't have a screen or anything. You basically had to already know how to do this new field of computer programming, but it enabled you to tinker and do interesting things for not very much money. And then just a couple of years later, they released the Apple II, which is a little bit more expensive, $5,500 to $11,000 2019 dollars. But again, cost operates our electricity, and they've made it easier. They've added on a couple of these modules, a graphical user interface, and sort of a way to move a cursor around a screen that makes it more intuitive to work with this thing. And so suddenly the skill barrier to work with computer engineering dropped at the same time that the actual cost of computers was simultaneously dropping. And then if we look on the software side, as this personal computing revolution was starting to kick off, there were companies that were starting to enclose the software landscape, the software commons, in creative monopolies, intellectual property, in this case, primarily patents and copyright. And there were a bunch of people who were there at the very beginning of computer science, computer and software engineering, who didn't like what they were seeing. They didn't like what Bill Gates was doing with Microsoft, basically. And they decided that they wanted to see if they could do something better and create an alternative to the IP enclosed systems that Apple and Microsoft were creating, and if they could instead create an operating system for computers that would be free and open for anybody to use. And so the product of that was the GNU operating system. So basically it's just a library of useful software for running a computer, and they sort of jujitsu'd the copyright restrictions by basically creating a copyright license for their software that made anyone who basically imposed on anyone who used them the obligation, the responsibility to freely share that software, that they couldn't enclose it, and that explicitly gave the users permission. You can do what you want with this. You can modify it, you can inspect it, you can run it on your computer, you can share it with your friends, you can even potentially commercialize it. Just make sure that any modifications and derivatives, you share it under this same permissive license, this open source software license, this GNU general public license. Excellent idea, excellent legal jujitsu. And then in the early 1990s, this library of useful pieces of software got combined with this sort of core computer running software called a kernel. Basically, Linus Torvalds and a few other people built this kernel for actually running, like the guts of the computer, and combined with GNU, it became GNU Linux, this fully open source software system. This was a thing that was developed more or less by, academics, hobbyists, people who were passionate about it, people who cared about computer engineering in their spare time, not almost entirely not for their jobs. Not for companies that they got paid by, and you would think, okay, a bunch of hobbyists, like, how are they going to compete with the titans that are Apple and Microsoft? Well, you look at it 30 years later, Linux is the core operating system of the World Wide Web. Pretty much every software company that runs web servers to serve their product, probably almost certainly including Zoom, the company that is streaming, enabling me to have this conversation with you right now, and that's recording this conversation. All of their web servers run Linux because it's just the best operating system for that, and it's the best operating system for that because it's so interoperable with basically anything else that you want to do, and it's been engineered by so many passionate software enthusiasts over the years that it just works really, really well for what they've designed it to do. And that really is a great inspiration to me. As you could imagine, it should be an inspiration to all of us, that like this thing that people who loved software engineering and who loved computers built out of love for it and out of a desire to share it with the world, became this core enabling infrastructure layer for the internet. Enabling not just the particular things that they wanted to do, but whole new industries. Just a lot of things. Very good. So questions that I want to try to talk about today, or the questions that we're going to try to answer today, are like, where are these equivalents for biotechnology? Where is biotech's GNU public license? This open source license that for sharing and for creating and growing the commons, the biotechnological commons. Where is biotech's Apple 1 and 2? I mean, Chaz has already talked to you about a core component of that Apple 1 and 2. And then where is biotech's GNU Linux? Where is the open source library of useful wetware that everybody can use? And so the first one, the open source wetware license, can answer that pretty quickly. It's Openmta. It's a thing that was developed by the Bioreactor Foundation, led by a really fantastic person named Linda Kale. It's basically that material transfer agreement I talked to you about. It's like the open source software license version of that, where you're still signing an agreement between the sender of the biological material and the recipient, but that agreement explicitly gives the recipient permission to copy, modify, redistribute, and commercialize the biotechnological materials they're receiving. And that's very, very important for starting to create this commons, this is enabling infrastructure for biotechnology, because that opens up the ability to share things with the world. So that's the open source wetware license. Then how about GNU Linux? How about the open source wetware library? So that is the Friendzymes project. So 4 years ago now, at the International Synthetic Biology Conference, SB 7.0, summer 2017, Drew Indy, who's one of the godfathers of synthetic biology, announced that the Biobricks Foundation had secured a large donation, and that they had basically decided to put that donation towards funding the synthesis and the free distribution under the Open Material Transfer Agreement of thousands of useful off-patent and or IP-free synthetic genetic parts. So basically, he went up on stage and announced, in so many words, we're going to make Linux but for DNA. And I got very excited about that because I was, staying up all night. The SB7 was in Singapore, and I was stuck in Ohio on an Air Force base at the time for a collaboration with my grad school work. And I got very excited because, like, yes, this is exactly what we need. This will break the wetware type stuff that IGEM has been developing out of that walled garden and enable it to be shared with the world. And so I reached out to Drew and was like, how can I help? And I learned that the project was actually not Drew's idea. The project was the brainchild of this biohacking prodigy named Keone Gandal, who has been doing bioengineering since he was in like middle school, and once bioengineered so good he got to meet President Obama. And so he gave a talk at the DEF CON hacking conference in Las Vegas back in like 2016, and Drew Indy was at that talk, and at that talk Keone basically said, we need a library of free synthetic genetic parts, or something to that effect. And Drew was like, oh, that's a good idea. And then he went out and fundraised for BioBrace and got enough money to actually do it. And so Keone wound up being the technical lead on the FreeGenes project for the first 3 or so years. And I became a Wetware developer, contributing useful parts to that. So that's FreeGenes. Oh, no, we're not going to do a tour of FreeGenes. Sorry, that was well, actually, you know what? Why not? Let's do it. Let's do a quick tour of FreeGenes if I can just find. Let's get out of that. And let's see. We'll just do a quick tour of FreeGenes because why not? Yeah, there we go. So this is the FreeGenes website. They've got a bunch of different Wetware collections around here that are already available. This is one I'll come back to in a little bit, but they've got a collection. If you want to bioengineer, this is something that Scott Ponal and led the development of, that I helped a little bit with. I will come back to that in a little bit. Collection for expressing proteins in E. coli. Apparently they've got a collection of parts for detecting COVID-19. So the important thing to notice here is there's all these toolkits that are available, like the molecular diagnostics toolkit, a bunch of genes for different like diagnostic enzymes and things like that. And the thing that you will notice is that this is kind of a store, and the cost of everything is $0. And so you can get all of the stuff you can find on this website for free. Basically, BioBrace Foundation will just prep it up and ship it to you for free. And so if you need FreeGenes, go to the FreeGenes website. Web license with the open source Wetware library. Then we're missing the Apple 1 and 2.
Isaac,
like the, what's that?
You freeze, I think, a little for the last 10 seconds or 5 seconds.
Okay, I'm gonna stop sharing and then start sharing again.
Okay.
Oops, sharing the wrong screen. Okay. Still sharing the wrong screen. All right. Desktop 2. Share. Keep clicking on desktop 2 and it keeps going to desktop 1 for some reason. Let's see. Maybe if I just directly go to all right, for some reason, it's only going to this one, to my first screen. So what I'm going to do is just slide this presentation over. Can you guys see the presentation now?
Not now.
No, can't see it.
I'm seeing the screen with the mountain.
Oh, okay. So it is actually on the right screen, just not showing it on my computer. Okay, cool.
Yeah.
You can see the presentation now?
Yeah.
All right. Pretend that didn't happen. Now, let's talk about Friendzymes. So, we got the open source Wetware license, we got the library, we've got the ability to build a library of open source Wetware parts. Now we got to build out, now we got to figure out how you put those things together along with low-cost hardware to actually build a functioning node of biotechnological production. A Frugal Biofoundry, a BioStack, whatever you want to call it. So our goal, we formed an IGEM team enabled by FreeGenes and the Openmta with the goal of democratizing recombinant protein production and purification, and also making it cheap and easy to engineer strains of cells, bacteria, yeast, various things like that. We got team members, last time I checked, in 9 countries on 5 continents, and we've got 4 collaborating labs, 3 community labs, and plus the Philippine Genome Center at Mindanao. Yeah, this is a nice map of various people on the team and the various locations that they're where they're at. We need to update this map because it is it has expanded. The team has expanded since then. So if we want to make, and I should clarify, why do you want to make enzymes? Well, enzymes are kind of the atomic workhorses. They're like little atom machines that they do all the work in biology. And so the ability to manufacture large amounts of specific enzymes and then purify them is at the heart of modern biotechnological industry, along with the more advanced techniques that involve just like whole cell engineering. And so the classical way that people have, particularly in like academic research labs, have manufactured particular proteins in cells is with E. coli. It's the most commonly used workhorse bacterial cell in research and possibly in industry as well. It's fantastic for cloning and handling DNA. It's a great organism for that, and it is basically, if you have the right equipment, it's pretty quick and easy to manufacture protein in it as well. The problem is that equipment is expensive. So E. coli doesn't really have a good secretion system. It's not very good at taking protein from inside of it and putting that protein outside of it. And so in order to get at the large amounts of protein that you're able to make inside of it, you have to basically pop the cells and then separate out all of the popped cell guts from the protein that you want. And in order to pop the cells, you need instruments like sonicators, which basically vibrate the cells to death with ultrasound, or you need like a French press, which pops them by like doing a rapid change in pressure through a tiny little hole. And then once you've popped them, you need large refrigerated centrifuges to separate those popped cell guts from the actual soluble proteins that you want. And all of these things, equipments for popping the cells, thousands of dollars, equipments for the refrigerated centrifuges, thousands or even tens of thousands of dollars. So that is not frugal. That is not democratized because it's expensive. And so what's another way of doing this? Well, our approach is to try changing the cell platform. So we could instead of using E. coli, we're looking at using Bacillus subtilis, which is a different bacterium, a gram-positive bacterium, single cell wall, great secretion system, very good at secreting proteins. And also Pichia pastoris, which is this yeast, which has a bunch of interesting properties. It's really good at growing on methanol, but it doesn't create ethanol as a byproduct, so it can grow to like super dense. You can get tons and tons of cells in your culture, so you can get high levels of protein. And both of them, importantly, very good at secreting enzymes. So we can use this secretion from these cells as part of our purification step, because we can try to secrete just the thing that we want, so that it's mostly just the thing that we want outside of the cells, and then we just like pellet the cells or clump the cells together and make them settle out, and then the thing that we want is left in the culture. Much simpler, much less expensive. And so in terms of wetware design, the things that some of the stuff that Friendzymes has been doing, we've been working on building a free and open source Bacillus subtilis genetic toolkit. So we need all the parts required to do to control gene expression in Bacillus subtilis. So that's the promoters that control transcription, terminators that tell us the RNA polymerase where transcription needs to stop. We need selection markers for Bacillus subtilis to make sure that once we knock a genetic part that we want into the cell, into the genome, that we can select for the cells that actually took it up properly. Homology arms, these are just pieces of DNA that are identical to particular parts of the genome, so that you can stick something directly right at that part, right at that point in the genome. And we also need to look at how do we get to high expression levels while avoiding some of these sort of in the weeds issues that happen when you're trying to get to high expression levels in Bacillus subtilis. You need multiple copies of the gene, but if you have multiple copies of the exact same DNA sequence right next to itself, then it can do this recombination thing and delete itself, and causes basically instability. It makes it so that the cell doesn't perform the way you want to in terms of manufacturing proteins. And quite similarly, I feel like this is the, oh, I see. Let me just really quickly swap over. This is, I was presenting from a, I was presenting from an older talk, that's why. Okay, cool. So on the bacillus side, you know, what have we done in terms of looking at how do we engineer this stuff? Well, the first thing we did was we called up Dan Ziegler, who was the former director for 30 years. He recently retired. He was the director of the Bacillus Genetics Stock Center. So one of the, basically the worldwide repository, if you wanted to get a strain of bacillus that had particular properties, you go to the Bacillus Genetics Stock Center. So he knows a lot about bacillus. And so he talked to us about, you know, what are some good, how do you choose the right strain of bacillus? You know, a strain that doesn't, for instance, secrete protein degrading enzymes like proteases outside of itself, because if a strain is secreting those proteases are going to chew up the protein you're trying to make and secrete into the extracellular medium. Should you choose a strain that can sporulate? You know, Bacillus cetullus is able to form these really hardy and really resistant spores that are nice for if you want to like share a strain, because then you don't need a cold chain to share. But if you want to manufacture something, a bunch of sporulating bacillus reduces the amount of protein that you can manufacture. You know, how do you find hypersecreting strains? All these, and how do you do competence? Like, how do you get strains that are good at actually taking up the synthetic DNA, the synthetic genetic devices you actually want to make? And so, with Dan Ziegler's help, we selected these IP-free strains that actually Dan Ziegler himself engineered and developed. K07, K07A, and K17S. Basically, they're called K07 because they knock out 7 of the extracellular proteases the bacillus normally produces, so they don't produce those proteases anymore, and therefore they won't chew up your recombinant protein that you're secreting out of the cells. K07A got an extra protease knocked out. K07S knocks out sporulation, so you don't have to deal with spores when you're manufacturing your proteins. And then, you know, a couple other things talking about, you know, how do you do proper DNA replication, and how do we want to turn the gene expression on, different promoters. So basically, Dan was extremely helpful and he also shared with us plasmid maps for like 20 useful bacillus plasmids that we could pull from. One thing that we're particularly proud of that we've done is started taking steps towards making it simpler and easier to screen for high secreting Bacillus cetullus strains, and screen for devices that secrete large amounts of the protein that you want out of Bacillus cetullus. So Bacillus cetullus naturally secretes about like 150 different proteins, and each of those proteins has this little thing called a signal peptide. It's a secretion tag, basically, that tells the cell secrete this protein. And there are 150 different ones of these. Actually, there are actually more of that from other Bacillus strains, but you know, for our purposes, we'll say there's roughly 150 or 170. And the issue you run into is that it is basically impossible to predict which secretion tag will efficiently secrete a given particular protein. You know, you know they work with the like original protein they evolved with, but if you're trying to put in a new protein, you basically have to screen all of them to try to find one or two that work well with your protein of interest and that secrete your protein of interest. And that's an issue because when you're building these genetic devices, you don't want to have to purify and then digest 170 different plasmids every time you're trying to build a new thing. That's not democratized. That's a big pain. That's a big pain to do. And so we developed this strategy, this Wetware design, to basically, instead of having to do 150 of those, we just consolidated all of the secretion tags onto 6 plasmids. And they're all flanked by these, you know, restriction enzyme cut sites. So basically, you introduce this particular enzyme, that's a particular pair of DNA scissors that recognizes a particular 6-base pair sequence. And that 6-base pair sequence is on each side of the secretion tags. And so it'll cut out all of the secretion tags from this long, you know, this tandem array of them. And so what you'll get is a pool of many, many secretion tags that all can be assembled as the same type of part into a bunch of different variants of the genetic device that you're building. And so in one pot, in one reaction, you can build many variants of the same device, basically varying just the secretion tag, and then you can transform that into bacillus, and hopefully we'll be able to screen for high secretion with like a fluorescent protein tag or something like that. So simplifying the process of developing good strains for making whatever you want. And then another important thing that we looked at is, all right, that problem of how do you make a lot of protein in bacillus? Because with E. coli, the way you make a lot of protein is you put the gene for the protein you want on like a high copy plasmid. Where a plasmid, basically a small piece of circular DNA that copies itself inside the cell and forms a lot of copies. And so once you have a lot of copies of that plasmid cell, a lot of copies of the gene and the promoter expressing it, so you basically all of the cell's resources when you turn on expression of your protein get directed towards manufacturing your protein of interest. With bacillus, it was more difficult because usually in bacillus people work with sort of basically directly integrating the gene they want into the genome. And in that case, you only have like one or a few copies of the thing that you want. And so the overall expression level is significantly lower. However, thanks to Keone, who's done a lot of sort of background research on bacillus, he directed us to this family of actually bacillus plasmids called the PHT family. And in particular, this variant PHT43, which has which can basically replicate in bacillus in a way that maintains the genetic stability of the plasmid, but that can also be engineered and propagated. The plasmid can be propagated in E. coli, and that's really great because E. coli is still undefeated as the best organism for propagating and growing up a whole bunch of plasmid. E. coli is the best for that, and so we can grow this plasmid in E. coli and get a whole bunch of it, and then we can transfer it into bacillus and it'll manufacture the protein we want. And because it's a replicating vector, it's able to produce pretty decent amounts of protein, like 10 to 15% of the total cell mass. It's a good amount. And so this is the original, sort of overall layout of the PHT43, but we decided to sort of refactor it and make it better. We ordered from T. Rather than that whole, plasmid, we broke the plasmid. That's the chlorophyll resistance gene. Yes.
Could you repeat the last, I think, 10 seconds?
That's 10 seconds. Oh, okay. Yes. So rather than just order this whole plasmid the way it is now, we thought, wouldn't it be nice to order a variant of this plasmid or to make it more modular and more engineerable? And the other reason that we decided to break it into parts is that 8,000 base pairs is too big to order from a DNA synthesis company for it to be very cheap. And so we broke that PHT43 plasmid into its component parts. For instance, this is the chloramphenicol resistance gene. This is the selection marker for bacillus. This is the thing that enables you to select for just the bacillus that have the plasmid inside them. This is CoLE1. This is the sort of origin of replication for E. coli. So that's the part that enables the plasmid to copy itself in E. coli. This is the LACI. This is the promoter that actually drives the expression of the protein that you want. That's, once it's inside bacillus, all these various things, E. coli selection marker. So these are all different parts, like this right here is, this whole thing is the origin of replication, everything that's required to replicate the plasmid in Bacillus subtilis. And actually, we were able to find a paper that shrinks this down a little bit just to this fragment that's more like 2,000 base pairs long instead of like 3,000. And so we're breaking this plasmid up into these functional parts. And we also want to add in a couple extra functional parts, these ribozyme insulators. These are basically little pieces of DNA where if you stick them in front of a gene, it just makes the gene behave more predictably. If you're trying to drive expression with a particular promoter, you stick an insulator there, then it's going to behave more consistently no matter what protein coding gene you stick after the insulator. Just makes things more programmable, more engineerable. This is what we want. This is what we want in bioengineering. And so added a part for ribosome insulator, added some parts for some transcriptional terminators. Again, these make the thing more engineerable and more predictable. And so we've broken it up. We've broken it apart. We've got these different parts, and then how do we put it back together? So we've created an expansion of a genetic assembly standard. So essentially, what we do is we define these particular short DNA sequences that are going to be overhangs. And when we add in our molecular scissors, in this case, the enzyme BSA1, it's going to cut out our parts, and it's going to leave behind these 4 base pair overhangs. And as long as we keep these overhangs consistent for a consistent type of part with a particular function, then it will assemble correctly into the plasmid. And so just to give an example of the types of parts that go into our expanded assembly standard, the types of parts you could slot in to a plasmid to do different things. You have the E. coli selection marker. So the thing that's the antibiotic resistance gene that allows the plasmid to propagate in E. coli, or that allows it to be selected for propagating E. coli. The E. coli origin of replication, that's what actually tells that's the machinery that allows the plasmid to be copied in E. coli. Then we've got homology arms, and optional. So this is the thing where if you want to integrate the genetic device into the genome of your target organism, like Bacillus, you need a 5' homology arm for that. Then you've got a target selection. So this is the selection marker for Bacillus subtilis that allows you to select for the cells that have taken it up. Then we've got these basically a bunch of other things. The bottom line here is that previous assembly standards only really focused on, all right, okay, let's have a promoter, let's have a ribosome binding site, let's have a coding sequence, protein coding sequence, and let's have a terminator. And there were a couple of assembly standards that added on a couple of tags onto the protein coding sequence, things like that. But what we have here is an assembly standard that basically allows you to build and modularly swap out entire plasmids, entire genetic devices from the library in a very powerful and modular way. And it's backwards compatible with the original MoClo assembly standard. So we're very excited about using this MoClo assembly standard. We have had a couple of issues in terms of synthesis. So the origin of replication for Bacillus subtilis has a bunch of repeated sequences in it. And so it's been hard for the DNA synthesis companies to make it. We're working with Keone to see if we can do a workaround there. But in the meantime, we're also designing and ordering homology arms to do genomic integration. And then Wetware design. So that was all Bacillus, but Bacillus is a bacterium, and while there are some things it's really good at manufacturing and secreting, there are other things that it's not. Like basically any mammalian or human protein, Bacillus will probably struggle to manufacture that. And so if you want to make other things, other useful stuff, the yeast Pichia pastoris is where we're looking at there. And as I mentioned before, and as I might just pop out to show you one more time. Yes, brief detour back to the Open Yeast collection on the Free Genes project. So we've got all these genes that Scott designed and a couple that I contributed. All these different promoters for controlling expression in brewer's yeast. So this is Saccharomyces cerevisiae. We also have promoters for controlling expression in Pichia pastoris. These are strong promoters for driving recombinant gene expression. We've got a bunch of different regulatory elements, terminators and things like that. And Scott designed with inspiration from I believe Duber Labs yeast toolkit. Scott designed these assembly connectors. So what's really beautiful about this assembly standard, and this is the same assembly standard that's used for the Open Yeast toolkit, is with these assembly connectors right here and right here, you're not limited to just expressing a single protein coding sequence. You can stitch together with these assembly connectors in a second assembly reaction multiple genes into a larger genetic device that, for instance, if you wanted to express 5 copies of the same protein, increase the copy number, increase the expression level, you could do that here. If you wanted to simultaneously express a restriction the molecular scissors, and a restriction methyl transferase, which is the thing that basically decorates the bacteria's genome with little modifications that make sure that the scissors don't shred the bacteria's own genome. You could do that with this assembly standard and with the linkers that Scott designed. And so I think that's lovely. It's going to be super fun and useful to use that. So that's all the wetware stuff that we've done so far. And then, once when we're starting to actually receive this DNA in our labs, and we're starting to, run various transformation protocols, start growing up the plasmids in E. coli, and start preparing to do actual assembly reactions for building these genetic devices, for building like PHT43 variants with our assembly standard. And how do we actually do that? For that, we actually need, high throughput strain engineering, and we need basically a Frugal Biofoundry. At the heart of the Biofoundry is, of course, an low-cost automated liquid handler, because otherwise you're going to have to, manually move small volumes of liquids back and forth over and over and over again to set up the genetic assembly reactions and the transformations and things like that. We don't want to do that. We don't want to make that a barrier that people have to learn in order to start participating in this. And so we're very gratified that Openmta donated the equipment, enabling basically all 4 of our collaborating labs to have fully decked out OT2 robots with P300, P20 multi-channel pipettes, and the mag deck, so we can do magnetic bead-based DNA purification, and the temperature deck, so we can run Golden Gate reactions right on the robot. And also, set up transformations and things like that. We're very excited about that. And then we're working on obtaining the means. So that basically handles the build side. We've got our wetware designed, and we're going to build it with the Openmtas, and also probably some manual setup in the early days. And then we need to test it to see if it works. So see if we built the DNA sequence that we think we built. We need to actually sequence things, and for that, we're looking at obtaining the Oxford Nanopore MinION DNA sequencer, which is, 25 times cheaper than the least expensive Illumina instrument, and has like $90 flow cells for that. If you barcoded your samples properly, you could probably get to maybe like less than $1 per plasmid sequencing would be my estimate. And then we also need to check to see, once we've transformed the DNA into a bacillus or pichia, is it actually manufacturing the thing that we want? And how do we do that cheaply? Well, you tag the protein that you're interested in with a fluorescent protein that glows, basically. And then you grow up many different variants of your strains in deep 96 well plates. You can grow them 96 at a time. And then you spin down those 96 well plates or flocculate them. And then you take off the liquid from them with your liquid handler, and you read them in a plate reader. So an open source microplate reader, a design exists that costs about $3,500 in components. We're looking at seeing if we can source some components from like original equipment manufacturers in China to bring that price down more, because I think we should be able to do that. And if we can build this instrument, then we can basically test the function of genetic devices 96 at a time pretty easily. More than that, because it's literally just like stick the plate on there, read it, get your answer, and you can do that for multiple plates. And so that's where you start to get to really high throughput testing of genetic devices that can match the throughput of the build on the Openmtas. And so that's something that we're really excited to keep working on. And actually, apparently, the open plate reader build is in the process of starting. So we raised about $1,000 on GoFundMe, and that seems like it's enough to at least start the build. And we'll look in with excitement and anticipation to see how Hive Biolab, which is one of our collaborating labs, builds out that plate reader. Yes, and here's where all the Openmtas are. They're at Open Science Network in Vancouver, in Chicago at BioBlaze, at Kumasi at Hive Bio, and Mindanao at the Philippine Genome Center. So that's sort of like how you get to a high producing strain. You do that high throughput design build test till you find a strain that's highly productive. And then once you have that strain, how do you sort of scale it up at least enough that you can make enough of, let's say, make enough of a laboratory enzyme that you never need to buy it again. For that, you need sort of frugal protein production and fermentation equipment. So the standard method for doing this, you grow up your culture in like a baffled glass shaker flask that mixes the culture in like a shaker incubator. Those glass flasks, tens to hundreds of dollars. I mean, it's okay, but it's not great. Tens or hundreds of dollars for some glass. You would prefer that it be cheaper. And then if you want to do like really highly productive fermentations, something that approximately would scale if you wanted it to, from like the 5 liter scale or the 1 liter scale all the way up to the 100 liter, the 1,000 liter scale. Then you need to start doing this with bioreactors, which are these instruments that basically, you put your culture in, you put your cells in, it mixes them up, and it aerates them, gives them lots of oxygen. It controls pH and temperature and various things like that to make the cells happy. And these instruments are expensive. They're thousands of dollars for the cheapest ones. They're tens of thousands of dollars for the more high-end ones. And so what are we going to try to do instead? Well, we are getting a lot of inspiration, a lot of advice from Sebastian Kochova, who is an absolutely wonderful independent scientist, community biotechnologist, biohacker extraordinaire, who lives in New York. And Sebastian has done various things related to frugal protein production, one of them demonstrating that 2 liter Pepsi bottles work quite well as shaker flasks for like 200 milliliter cultures, which is great for just sort of smaller scale prototyping. And he's also developed this frugal bioreactor that design that starts with basically just uses a big plastic carboy that's sort of skinny, an aquarium pump that simultaneously mixes and oxygenates the culture. And so the total cost of this thing is maybe $180. Apparently you could probably get cheaper if you got some of the equipment from like original equipment manufacturers in China, with a 4 liter reactor volume. And you can, at least with E. coli, which is the thing that Sebastian's tested it with, you can grow up just absolute scads of your protein of interest, just massive amounts of it. So we're very excited to try that. And in fact, one of Friendzymes team members, Diego Munoz in Peru, he is basically building a modified version of that frugal bioreactor, swapping out a couple of the electronic parts, swapping out the carboy for this container right here. And he's already got it temperature controlled. So we're excited to see how this develops once he gets the aquarium pump set up and start seeing how many cells you can grow, what kind of a cell pellet you can develop. So then once you've manufactured a bunch of your protein in your frugal bioreactor, then you need to actually purify it if you want to do more high-end things with it. There's some work that's been done that's really cool by like Andy Allington's lab, just not purifying things at all and just like doing things with like cellular reagents where you basically grow up the protein inside E. coli and then just like dry down the E. coli and then just like take a little on some paper and then just like punch out a little bit of paper, put it in your reaction, and then it just runs your reaction because it contains the enzymes. But if you want to do things like quantitatively, if you want to do things in an automated way, I think still purified enzymes are and in particular, if you want to start moving up the value stack for biotechnological products from, just sort of like reagent grade molecular biology enzymes, which is where we're going to start. If you want to start moving up towards higher and higher purity, higher and higher value products, potentially getting towards biopharmaceuticals, for instance, then you're going to need purification methods. And so for thermostable enzymes, like the sort of first target enzyme we're manufacturing, which is not fusion. It's fusion is trademarked by some other company. It's PFU7 to PFU SSO7D. So it's PFU DNA polymerase fused to this non-specific single-strand SSO7D DNA binding domain, It's based off the same design as Fusion, but Fusion's patent expired last year, so we can just do whatever we want with it. With thermostable enzymes, you can just basically heat up the culture. Heating up the culture pops the cells, but the thermostable enzymes, they're thermostable, and so they stay in the media while everything else sort of crashes out. And so it's a nice way of doing an initial purification step on the cheap. But if we want to get sort of higher purification, we're looking at doing dialysis. So this is basically just putting the liquid that contains the crude liquid that contains our enzymes of interest in a little basically sausage case, a tube with a bunch of little tiny holes in the walls of the tube, holes that are small enough to allow water molecules and salt molecules and smaller protein molecules potentially to exchange out into a larger tub, but that retain our protein of interest. And this is pretty scalable, pretty cheap. You can get like 38 of these potato chip clips that'll clip off the ends of a dialysis tube for $11. We should be able to do that pretty cheaply. And then for high purity applications, our mentor Dushanth Sivaratnam, who we talked to at the beginning of this year, he is a recently graduated PhD candidate from, so he's now just a PhD from the UK, and he has developed methods for basically extracting silica from sand with like acid and then precipitating it back out into these little silica microparticles that have really high surface area. And then you just tag your proteins of interest with silica binding tags, which exist. There are various organisms in the wild that need to bind to silica, like diatoms that have silica shells. And so you just take a little short peptide that binds really tightly to silica, tag that onto your protein of interest, run that protein through your really cheap extracted silica column, and you get your protein sticks to the silica, everything else flows through, and you've done a pretty decent purification on the cheap. Other cheap methods of purification, there are these things called elastin-like polypeptide tags, which are off patent now as of 2021. Basically, they're slightly larger tags. The main thing you do, you heat the culture up a little bit, or you add some salt to it, and the tags reversibly clump together and aggregate. And so you can turn the protein that you want into this aggregate that settles down, take off the supernatant, and then cool the culture down, or dilute the salt out, and it'll resuspend. And so it's another simple purification and concentration method. So we're looking at both of those once we actually get protein manufacturing going. And so that brings us up to where we are right now. And so now we're doing this hackathon. So this is showing you our methodology for the sort of wetware, hardware, software, and the lawware fronts. And hope this has been inspiring. But this is very much, there's a lot of work to do, and it's very much a collaborative effort. We collaboration with Joel, Just One Giant Lab, fantastic, meta community lab all around the world. We're hosting a hackathon that'll tackle how do we actually implement some of these frugal biofoundries? Can we make plans to build these frugal biofoundries, or to tackle particular components of this challenge all around the world? And so this event is going to be split into 3 parts on Saturday and Sunday: Asia, Oceania, the Americas, Europe, and Africa. Really looking forward to seeing you all there, and we're very excited to collaborate with you all. And I think that is everything I have to say. So I'm going to stop sharing my screen.
Thank you, Isaac. I think it's the more detailed version of our project, and I think this brings a lot of interesting ideas that people could use. And I would like to start the Q&A session. So anyone has questions for Isaac? I could start one. Someone. Okay, so I share the Friendzymes project with people in Brazil, and they are actually very interested in the project as usual. So people actually was like, "Oh, this looks very good," but they the PI, the most old person that have a lot of experience in genetic engineering, ask us this question. So how we could help you? I think I think most of them are very interested. And I think one of the first questions that they have is that how I could help this project. And only to finalize this is that they they say to me that they are very happy because Friendzymes share a lot of ideas that people in the 80s, in the start of the genetic engineering, have, that is build things together, collaborate in collabs actually. So so what how they could help? It's
Yeah, so many ways. I mean, I think that the biggest value add for particularly academic researchers that already have labs is in actually building and testing the wetware libraries that we've designed. You know, because it's probably going to take us a while to raise the funds to and to, you know, solve the sort of technical challenges of integrating the Frugal hardware side. But you know, if we have people who want to collaborate who already have the wetware skills to, you know, set up genetic assembly reactions and transformations and, you know, do the biological design build test cycle themselves, and they already have the equipment required to, you know, sequence the thing they built, or, you know, they have plate readers that can measure the fluorescence. And I mean, they don't even need plate readers because they've probably got, you know, they've got like instruments for SDS page where they could just do, you know, they could just look at what the proteins are that are coming off the supernatant. They can even look at, you know, purification. They probably have, you know, fast protein liquid chromatography instruments. So basically, almost everything that we've done up till now has been on the design side, both in terms of wetware and software. And you know, there are good reasons for that. Basically, that's the side that's most accessible because it doesn't require capital and skilled labor to integrate. And so now that we're starting to get into the hardware side, you know, there's going to be teams around the world that are working on building out the Frugal hardware. But at the same time, it'd be really great to start testing out the open source wetware.
Oh, Devin, could
Hey,
say anything?
thanks. Yeah, let me lower my hand here. That was a fantastic talk. I really enjoyed it, and I think it's great to be able to try and make these enzymes more affordable. So you mentioned that you need some people maybe to test this out that have the facilities.
Yes.
I teach biochemistry at a small college in mid-Michigan, and we have a lab, and I mentor an IGEM team here as well. I would love to test out some of these things. You know, some larger universities, maybe they can not even think twice about ordering some more enzyme. And, you know, we certainly take advantage of that when we can, but I think having a cheaper source for these things would really help for budgets at places like where I am, at high schools, at community colleges, and they're also the act of actually purifying these enzymes. These would make great laboratory activities for biochemistry classes. So yeah, whatever you're thinking in terms of how to test these things out, I'll throw my my email address in the chat here. You know, I'm going to be on the Slack channel and everything too, but let me know. I'll be happy to test them out.
Yes, you know, there's a component here, like one of the immediate applications, like the best possible applications for this project would be education, because you know, if you make it cheap enough to do bioengineering, then you make it possible for many more like teachers, you know, professors like yourself to actually, you know, get students' hands on this stuff, actually doing it. And that, you know, that makes me think of like the sort of distributed citizen science initiatives where, you know, instead of starting with like the lab robot that sets up everything, it becomes like the class is how do you build genetic devices, how do you set up these assembly reactions and transfer them and test them. And so like the entire class is like testing out, building out different variants of these parts and trying to find the ones that manufacture and secrete the enzymes most efficiently.
Yeah, I mean, you have need variants created or tested. I'm there. So I think you said it's a great activity for a classroom.
Fantastic.
I'd like to also add to this conversation that I think one of the purpose of the hackathon is to find out people like Devin that could actually be part of this network. I think Isaac already have cited sometimes before that we need to have this decentralized network of people producing reagents, producing enzymes, where they could share these things together, right? And one of the main ideas of the hackathon is actually to see people that have the same necessities, and we really want a frugal biofoundry, and they really want to have cheaper enzymes. So I imagine that other people that will be watching this or are in this meeting are looking for the same thing, and be free to contact us and participate of the Friendzymes.
Yes, and actually,
Yeah.
I want to add another thought that I had from the question you asked earlier, Isaac G, about how people can contribute and collaborate. Another thing that academic researchers have access to potentially is university budgets. And so, you know, it's not just on, you know, it's definitely, you know, with the existing capital they have in terms of the equipment they have, they can start testing out the wetware we've designed immediately. But also, you know, if for instance, Devin went to his university and said, I want to build this new class around, you know, build a biofoundry, and, you know, can you give me $20,000 to get, you know, all the robots and equipment that I need to set up a biofoundry with my class. Then suddenly you have a biofoundry, and once you have that, like the productive capacity of this Frugal biofoundry is theoretically high enough that, you know, even people who are like in far-flung places who just are not, it's going to be really difficult for them to get access to the resources required to build their own. They can design wetware and through this network, you know, basically get the DNA for free, and then collaborate with Devin and his biofoundry on actually building it out and testing it. And so, I guess I would very much say the academics and people who have access to capital that would be required to buy the components of a Frugal biofoundry, even if you just buy it and build it for, you know, your local area, you can still be enabling everyone around the world to do bioengineering.
Yeah, so Devin, it's good to meet you. I'm not sure how you got connected. We're all connected in strange ways on this group, but I'm Sarah Ware in the Chicago area, and I actually have taught a class at Benedictine University that I developed called Special Topics Biohacking as a university class for a bio elective. So I would be definitely happy to talk to you about that, and teach I've developed a microbiology class, all completely open source resources, no textbooks, no lab books needed. So both an online lab version and a version where you go in for labs, but I'm happy to definitely share resources with you.
Oh, great. That sounds excellent. I've also taught some sort of self-developed synthetic biology courses here. I'm at Alma College. We're a small college in mid-Michigan. And likewise, I'd be happy to share that with you as well. But yeah,
Okay.
however you want to share that or email me, or I'm just like all these different sites now, right? So whichever
Yeah,
way.
I'm putting my general email in the chat as well.
Excellent.
I'm on the Discord.
Okay.
Sorry, Chaz, for
Oh, Chaz, I know you got your hand up like a normal person. Sorry.
No, that's okay.
It's all good. It's all good. Yeah, I had a question, but you know, it's kind of on a different train of thought. So Isaac, I really enjoyed your chat, and you know, when I was talking, I kind of gave my background and how I was so interested in policy, but felt like policy had its limitations. So I went to learn how to develop the tools, and then you started your talk saying like, hey, the tools will only get so far, and you gave the great example actually of, I think, the COVID-19 pandemic response. So policy clearly plays a really big role in all of this, right? So I would love to hear your thoughts on you know, just the current policies that might affect this Biofoundry idea, and how they the Biofoundry kind of fits into the world of policy.
Yeah. So, you know, the way I think about this is it's basically like 1939 in the world again. We're facing an existential threat that, you know, that the way that threat should be responded to is with a massive and total mobilization of our economic and societal resources. We're headed for a very bad future if we don't do that and mobilize our resources in the direction of, you know, decarbonizing our energy material infrastructure and building sustainable energy and material systems everywhere on Earth. And, you know, there's no way that like the free market is going to do that in time. It's something that requires the level of capital and manpower, you know, people power that is available to sovereign governments, as international states. And so, you know, I spent the last 2 years of grad school basically alternating between trying to finish my thesis and getting increasingly involved in the Sunrise Movement, which is this organization in the US, the youth-led movement, mobilizing for this thing called the Green New Deal, like massive World War II scale economic mobilization of resources to decarbonize, build a good future with the just transition. You know, the only reason that I stopped doing that is because I graduated and had to get a job and had to pay a mortgage. So it's basically I'm doing this because this is what I can do. This is like, the two are very much necessary simultaneously. You know, the sort of the way I think about it is we need that mobilization of public resources and public capital. The democratization of technology makes that public capital go further. It makes that public mobilization easier, and it also makes it so that we're not dependent on like a single. Hopefully, we're not dependent on like the government of a single dominant superpower to take these steps. Like if we can democratize this and get bioeconomies going in all the countries in the global south, which are going to be hit first and hardest by, which are being hit first and hardest by the impacts of the climate crisis. You know, maybe they can probably mobilize faster than the countries that got rich by burning tons of fossil fuels and are still the heart of the fossil fuel industry and economy. So yeah, there's a lot there. I mean, so I've done a little bit of policy work as well for like the think tank New Consensus, and so I'd be happy to talk more later about you know, ways biotechnology can contribute to the good future and ways that mobilizing for the good future prompts us to rethink how we do biotechnology.
Yeah, very interesting. And yeah, would definitely love to connect and chat more.
So any other questions? Okay. Yeah, I think we finished the final meeting for the hackathon. We are really excited to see all the projects, all the ideas that people will bring to us. and present, and also try to see how we could help them to actually make this real. Actually, so thank you, Isaac, for your presentation, and for everyone to be here and spend some time thinking about synthetic biology.
Thanks, Isaac, Chaz.
So we'll be waiting you guys in the sessions that will be, I think we'll start the first session, the as if session, in the next 3 hours, maybe. So see you later. Goodbye.
Thanks for coming.
Bye.